Hello and welcome to the 16th episode of the Innova Biosciences webinar series. Today's webinar focuses on gold nanoparticles, optimization of conjugates, and the importance of size, shape, and surface properties in different applications. Our speaker today is Dr. Nick G., the CEO and CSO of Innova Biosciences. Today, Nick will outline how to determine the size and shape of gold nanoparticles, how to make conjugates using passive binding and covalent conjugation techniques, and the shape and size requirements of different applications. There will be a question and answer session at the end of the presentation. Please submit your questions by typing in the questions box in your control panel, which is circled in red here. And feel free to send questions throughout the webinar, as we will answer as many as we can in the Q&A session. And any outstanding questions will be answered by email in the next few days. So that's the basics laid out. Now please welcome Nick.
Okay, thanks Claudina. Here we have samples of 5 nanoparticles on the right hand side, all measured at 40 nanometer size, but you can see quite clearly that they're different from one another. Equally, this set of 5 samples is very different in color from bulk gold in the form of gold bars on the left hand side. So why do we see the red color, and why are there different shades? Okay, the reason is that gold nanoparticles can absorb certain wavelengths from white light. This is why you see usually a red color. And the energy from the light causes the surface electrons to oscillate, creating net positive and net negative charge. And this phenomenon is called Plasmon resonance. And it explains the color, and it also results in an electric field because you have these positive and negative charges. And this explains many of the properties, many of the useful properties of gold nanoparticles. Okay, the different shades can be explained in terms of different shapes. So the interaction with the light is altered when the shape of the particle is changed. So here we have transmission electron micrographs of the various samples of gold nanoparticles. And you can see in the bottom right corner some spherical particles, and there are other shapes and sizes. So in the bottom left you can see a triangular structure, and if you look around the slide you can see rods and some hexagonal particles as well. And in the top right you have this structure, looks to be a fusion of maybe 5 or 6 nanoparticles. So TEM is a fantastic technique for looking at the shape of individual particles, and it explains why you see different shades. So particles that are all said to be 40 nanometer can still look different because of different shapes. Okay, moving on. Now, not everyone has access to TEM, but you can tell an awful lot about gold particles just from what your eye tells you, and also simple absorbance scanning in the range 400 to 700 nanometers, which is shown here on the left. So a well-dispersed suspension of nanoparticles will give the quite a sharp peak, as shown here with the black curve, and in the presence of salt the whole system is destabilized and you get aggregation. So you can see that by eye quite clearly in the image on the right, and the absorbance profile changes completely. The green line shown here. In particular, there's a huge increase in absorbance between wavelength 600 and 700 nanometers. So the ratio of the absorbance at 650 to 530 is a useful aggregation parameter. You can also tell more about how spherical the particles are from the absorbance scan. So those familiar with chromatography have perhaps heard of peak width at half height. We can't quite use the half-light method with gold nanoparticles because sometimes you can't get the reading at the 50% level. So, but by just using peak width at 75% of the y-axis, you can measure the distance between the peak going upwards and then coming downwards. So in this particular case, we have peak width at 75% height of about 45 nanometers. The monodispersed spherical particles will also have a low 650 to 530 ratio. The additional information you get from such an absorption profile is a crude measure of particle diameter. So the peak absorbance for 40 nanometer gold in water is about 526. So if you see a profile like this with a peak at around 526, it's probably around 40 nanometer. This is not as reliable as the electron micrographs. You know, you're looking at a population here, but it gives you a rough indication of the size. Okay, if you see a profile for any solution of nanoparticles, gold nanoparticles, the profile will look vaguely similar, but when you overlay them, you can actually see that there are significant differences. So again, the samples that you saw earlier, here's some absorbance profiles, and you can see that they vary in width at 75% absorbance, and they also vary in the region of 600 to 700 nanometers on the x-axis. So the sample which has the highest reading at 650 is probably showing signs of some aggregation because the curve's lifted off the baseline, and the others don't really show much aggregation, but you can see they're different in terms of peak width, and this relates to the shape and really is consistent with what the electron micrographs tell us. So absorbance profiles, really easy to get, tell you a lot of information, useful information, not quite as good as TEM. It's great if you can do both, but still very valuable technique. In this particular case, the narrowest peak width we have is 45.5, and most of the samples are below 0.2. With a really good batch of particles, you might be getting around to 0.1 for the 655-30 ratio. Now, for different applications, you may need different shapes. So this is not a question of good and bad necessarily. Different types of nanoparticles with different shapes might work in different applications. I'll come on to that more later in the presentation. Okay, I'm going to turn now to methods of attaching molecules to gold nanoparticles, and whatever the size and whatever the shape, these approaches are valid. So we have two types of material. We could have naked gold, which is gold that doesn't have anything on the surface other than counterions, and it's usually citrate, which adds a bit of negative charge and helps to maintain colloidal stability. Completely different type of material is gold to which you've attached some sort of surface layer, and I'll come on to that later and what the advantages of doing that might be. So we have two materials, and there are three basic approaches to conjugation. So for small thiolated molecules, we can use an approach called self-assembly. This is almost covalent, but not as strong as a typical covalent bond. We have passive adsorption of proteins onto naked gold, and we can also use covalent attachment, either of small molecules or large biomolecules, to coated gold. So I'm going to come on to each of those in turn. So the self-assembly technique has been used for years with planar gold surfaces, and the idea is you exploit the high affinity of thiols for gold metal, So if you have a molecule with a thiol group and some other functional group, say X, at the other end of the molecule, you can actually create a metal surface which displays the functional group X, because the thiols will automatically assemble in an orderly fashion on the gold surface. Now this approach can also be used with nanoparticles, and so it's described here. So here we have the naked gold with the stabilizing citrate ions. They're only very loosely absorbed onto the gold surface. The thiolated molecule, the thiol represented by the blue blob, is added, and hopefully we get a structure like the one shown on the right here, with the thiols arranged in an orderly fashion, and then whatever is at the other end of the molecule, a functional group, or it might be a drug, whatever it is, it's displayed on the surface. Now, one of the disadvantages, but maybe advantages in certain circumstances, is that the thiolated molecules can dissociate, because each molecule is anchored by a single point, and if there's a dissociation event, then the molecule will essentially fall off. What happens then is you have bare metal exposed, and the particles can aggregate as shown at the bottom, so there's a change of color and the whole thing turns into a mess. Now generally that's bad, but if you actually want molecules to dissociate, and there may be sort of therapeutic applications here, then this is potentially a useful technique in that the molecule's not firmly anchored. Okay, if you use this approach and the rate of dissociation is too high, what you can do, as shown on the right, is to introduce two thiols into your small molecule, and this promotes a stronger interaction with the gold surface. And you can take this further, you can do 3 or 4, and then it's then down to the chemistry. Can you actually incorporate so many thiols into a small molecule? And again, as shown here, X could be a drug, or it could be a functional group, say carboxyl, which you might use to carry out further conjugation reactions. But the system generally is a little unstable, but you can improve it perhaps by introducing multiple thiols. Okay, turning on now to the second approach. This is attachment of antibodies or large proteins to naked gold. Now there's a high risk of aggregation with this approach if the conditions are not quite right. The binding of the antibody is pH and salt dependent, and it's often said that the best pH is equivalent to the isoelectric point of the antibody plus 0.5. So for example, if the isoelectric point is 8, you should perhaps do the binding at 8.5. Now usually you don't know the isoelectric point, and in practice the way that conjugations are carried out is to use a range of pHs, and I'll give you an example of that on the next slide. The binding mechanism is not completely understood. You need to carry out multiple trial conjugations to get things exactly right, and at the end, after you've blocked the gold surface with BSA, after you've attached the antibody, just to make sure there's no bare metal, you perform a stress test with sodium chloride. As I mentioned earlier, if you add sodium chloride to naked gold, the gold particles will aggregate instantly. So this is essentially a test of whether you have any bare metal. And if you have, the sodium chloride will drive aggregation. Okay, before I come on to the actual examples of conjugation using passive adsorption, these are the particles that are used in the next slide. So this is gold that we produce here. The electron micrograph shown on the right. So these are nice spherical particles. Consistent with that, the peak width at 75% height is 43 nanometers. We also have quite a low 650-530 ratio. So using these particles with 4 anti-troponin antibodies, we've determined the best conditions for each one. So what we have here is, so this is a salt stability test. So we're showing on the y-axis 650-530 ratio of absorbances, and on the x-axis this is the amount of antibody that we're using to try and make the conjugate. And the whole thing is carried out at 5 different pHs. So to optimize each antibody, there's quite a lot of experiments that need to be done. It's a trial and error procedure. So in this case, top left, this particular antibody seems to work best at pH 8. And what I mean by that is we have a very low ratio with the blue line, for example, and we're using quite a small amount of antibody, 3 micrograms in this case, to efficiently coat the gold surface. For the antibody 2, top right, the pH optimum here is quite different. It's pH 6 or pH 7, both look good, but the profile in general is very different from that for antibody 1. Going to bottom left, this time it's pH 6 optimum, and bottom right, this is significantly different from the other profiles. pH 7, 8 or 9 seem to be giving conjugates with very high salt stability. So using this is a tried and trusted method. It's just carried out systematically, and in all 4 cases here we were able to get stable conjugates. Okay, turning on to a covalent approach to conjugation. Now this is radically different from the method just described, but it's significantly easier. So let me explain what we have here. So this is an electron micrograph. In the bottom sector, you can this is a support grid that you can see, and the support grid has a tiny hole in it, which is at the sort of right top left area. And what we have here is a coated particle. So we have a coating material that's been wrapped around the nanoparticle, and this particular particle is half on the grid and half off. And in the area where there's a hole, you can see the surface layer quite clearly. It's about 1.5 to 2 nanometers thick, and this coat completely changes the properties of the gold particle, because the metal surface is not facing the external environment now. It's completely shielded by the coat. So we can see the effect of the coat here, again using various stress tests. So on the left hand side, for comparison, we have naked gold, and we have the nice peak for the profile in water, and then in the green, blue and red curves we've got sodium chloride 1 molar, or hydrochloric acid, or sodium hydroxide. And in these conditions, gold nanoparticles will aggregate instantly, and you get the profile with the absorbance ratio 650 to 530 is usually around 1. Now you can see on the right hand picture with the coated gold, the coated particle is now completely resistant to salt, it's resistant to acid, it's resistant to sodium hydroxide. There's no aggregation whatsoever. All 4 curves are superimposable. So the coat shields the metal from the external environment. It becomes very resistant to the usual stress tests, and in fact this material will survive 2.5 molar sodium hydroxide at 70 degrees C for 90 minutes, and that's the basis of our QC test for this material. I should point out that this material cannot be used for passive binding, because the antibody cannot actually make contact with bare metal. So this is just for covalent conjugation. Okay, these are some of the materials that we have. So here again, electron micrograph of the particle, and we've just annotated it with various functional groups. So starting from top left, amine, carboxyl, maleimide, biotin hydrazide, streptavidin, provides a whole range of possible chemistries that you can use to attach small molecules or antibodies. So for example, with the amines, you can attach any NHS ester to the surface coat, or you could use a carboxylated molecule in combination with a carbonamide. And then top right, the carboxyl, similar reaction with a carbonamide, but this time you would have an aminated molecule. Potentially a lysine from a protein molecule, for example. And then, with maleimide, you can attach thiol-containing molecules. Bottom left, hydrazide will react with molecules with aldehydes. And then we have derivatives with biotin and streptavidin, and they will obviously bind the appropriate streptavidin or biotin labeled molecules. So not covalent in that time, but in all cases, irreversible attachments of analytes and antibodies to a gold particle. So in the case of antibodies, because this is so commonly done, we've developed a special one-step conjugation kit for antibodies. It's analogous in some ways to Lightning Link for those who are familiar with that product of ours. So because we have this surface coat, we can actually freeze dry the coated gold, and we can add in all the other chemistry in a freeze-dried format that's required for a conjugation reaction. So the conjugate can be prepared simply by pipetting in a solution of the antibody. It reconstitutes the freeze-dried mixture. The reaction just happens, ready to use in 20 minutes, but your hands-on time is just the pipetting step. So really easy, but there's all sorts of other chemistries available if you want to couple other molecules with a variety of groups. Okay, so passive binding versus covalent attachment, which is the better approach? Well, it depends. There's no right answer here. I think it's good to have a variety of options. So with naked gold, you can couple whole antibodies with the traditional passive approach. I showed you the data for that. It's a trusted approach, very reliable, but very time consuming. You can also couple small thiolated ligands. We showed some examples of the self-assembly technique. Particularly good if you want to exchange those small molecules on the surface, or if you want them to fall off. And by attaching a surface coat we call innovaCoat to the particle on the right-hand side, then again you can do whole antibodies. It becomes much easier than the passive binding method. You can also do small molecules with a truly irreversible link. Also useful for small antibody fragments, which sometimes unravel on a bare metal surface. With the innovaCoat derivative, the antibody fragment will not actually make contact with metal. If you want molecules to fall off in a controlled fashion, you could incorporate a cleavable link. So the molecule stays on, it only comes off when you want it to come off. As I mentioned, there are multiple chemistries, and at least for antibody conjugates, you generally require less antibody with the coated particle, and that's perhaps because less of the antibody denatures, because the antibody doesn't touch the metal surface. Okay, this is just an example. This is lateral flow data that we're showing here with some CRP conjugates, and the data that you see is for covalent conjugates made with the innovaCoat GOLD. So the pairs of conjugates for each antibody, CRP1, 2, and 3. And we can see that we have a strong signal in all cases, and the level of signal obtained with a passive conjugate is indicated by the horizontal black bar. So, so for the left-hand pairs of conjugates, CRP1 and 2, the performance is essentially the same regardless of whether you use the passive binding method or the covalent method. Covalent method is much faster, so that might be a consideration. But with CRP antibody 3, the covalent approach actually gave a much higher signal than the passive method. I'm sure if we did enough antibodies, we would find one that would be damaged by covalent modifications. So I think it's great to have both approaches, and it's just a question of trying both, but perhaps starting with the covalent because it's so simple, and if that delivers the performance you need, then you don't have to get into all the pH and salt titrations of the passive method. Okay, the other conjugation methods that can be applied to coated gold is oriented coupling. In this case, we're showing periodate treated antibody. The periodate positions aldehyde groups on the Fc domain, and this can react with the hydroxide derivative of gold, thus projecting the antibody binding arms out in space, hopefully favorably positioned for the interaction with the antigen. You can also use the gold for coated gold for oriented coupling of antibody fragments, for example FAB primes. So the thiol groups shown here will react with the maleimide very efficiently, and you can get a fragment on a gold particle without the protein metal contact. You can obviously use exactly the same fragment and use the self-assembly approach using bare metal nanoparticles, but sometimes that damages the fragment, and it's usually better to use a surface coated particle if you've got small fragments. Here's some data just to show the benefits of orientation. Here we see a fragment oriented through the cysteine versus a random attachment through lysine to a carboxylated surface coat. So in this particular case, you see additional performance because the antibodies presumably are oriented more favorably with the cysteine maleimide link. Okay, so just a summary of the various nanoparticle tools that are available and the conjugation methods. So gold nanoparticles come in many different shapes and sizes. And the example I showed you, 40 nanometer gold, although they're all 40 nanometer, you have lots of different shapes, different colors, and different properties. You can attach molecules to gold by passive absorption, self-assembly technique, or you can irreversibly anchor molecules if you've got a surface coat. In the case of antibodies, there's a simple one-step covalent method available now, but there's a whole range of covalent chemistries for other analytes, and depending on which functional group you have, you'll need a particular type of coated gold. The best approach in any situation is going to depend on the nature of the substance that you want to conjugate and the intended application. And it's just a question of screening. It's a screening exercise really to find the best combination for your particular antibody or small molecule. Okay, I'll just remind you here of the properties of gold nanoparticles, which stem from the absorption of energy from white light, causing this oscillation, this Plasmon resonance, which gives, so you get the color, which allows gold to be used for detection, and you also get an electric field created, and this field is altered by the environment, the type of solvent that the particle is in. Usually it's water or a water-based buffer. Also affected by other molecules or other particles in close proximity. So these features of gold have been exploited in a number of different assay formats. Okay, here's a snapshot of applications. So two areas really, detection or therapeutics. So on the detection side here, major use of gold in lateral flow, surface Plasmon shift assays, surface enhanced Raman scattering or SIRS, metal enhanced fluorescence, and microscopy. In terms of therapeutics, phototherapy, radiofrequency ablation, and drug delivery and targeting. I'm just going to pick out some of these methods now, tell you a little bit more about those. Okay, Plasmon shift assays. So what we have here is a demonstration of a Plasmon shift using streptavidin attached to coated gold and biotin attached to coated gold. So the absorbance profiles for those two entities, the separate entities, are shown in the red and blue. So you can see a sharp peak and a low 650 to 530 ratio for those curves. So essentially these particles would appear as red. And then if you mix the two together, obviously the biotin and streptavidin particles will associate with one another. So the peak actually moves position, shifts to the right, because you now have something that's of greater size. But the main change to the profile is the uplift in absorbance of the green line on the right hand side in the 600 to 700 region of the x-axis. So this is the basis of an assay where you can monitor interactions of biomolecules. In this case we've just used two particles with binding entities. So same materials, just a different experiment. So this is looking at the time course. So the blue line shows the increase in absorbance at, in this case, 620 nanometers. And you can see this by eye, so it can be a simple readout in a plate. So the image here shows with competing biotin, so the particles stay separate and appear red, and then without biotin, the particles aggregate and you get the blue color. So you could use this obviously as a biosensing assay for biotin. So this is just for illustration, but you can think of any number of analytes that you could use with this setup if you just had the appropriate binding entity attached to one of the gold particles. Okay, SIRS, surface enhanced Raman spectroscopy. This technique has been used by chemists for years. It provides information about molecular vibrations, and that tells you about structure. But it can also be used by biologists and to look at interactions between molecules. So a Raman dye is analogous in a way to a fluorescent dye. It's a reporter molecule. Usually the Raman signal from a molecule is extremely weak, but if you position the Raman dye in the electric field created around the particle by the Plasmon resonance, and the field is particularly intense when you have two particles close together, or you have a rough surface, or you've deposited nanoparticles on a surface, the Raman signal, when it's from a Raman dye, when it's in this electric field and very close to the surface, can be amplified by many orders of magnitude. So potentially the Raman dyes can be used as labels in conjunction with gold particles. So this is a hot area, and I think we'll see biologists using this increasingly in the future. Okay, so we've got two things here: fret quenching and metal enhanced fluorescence. And I can mention Raman again briefly. So if we have a fluorescent molecule here, the green molecule, and it's moving towards the gold nanoparticle. Now, if the fluorescent molecule attaches directly onto the metal surface, although it's a fluorescent molecule, it will be quenched in that situation. But you might see this Raman effect, a massive amplification of the Raman signal. If the molecule happens to be positioned between 0 and 5 nanometers from the surface, then you get a quenching effect, and you can see how you could use this to study biomolecular interactions. So the fluorescent molecule at distance from the particle gives a bright signal, and it's quenched if there's an interaction with the particle. Equally, if the fluorescent molecule is positioned 10 to 30 nanometers from the surface, this is particularly seen with silver but also with gold, then you can get enhanced fluorescence. So again, you can see how you could use this to study interactions between gold particles and other molecules, where you either get amplification of the Raman signal, you either get fluorescence quenching, or you get fluorescence enhancement. And this is all to do with the electric field that's created around the gold particle. It leads to all these possibilities. Okay, coming on to some of the therapeutic applications now. Photothermal therapy. Now, this is an interesting approach. You tend not to use gold spheres here. It tends to be rods or hollow spheres, or sometimes spheres that have a thick silica shell. The reason these shapes are used is that the absorbance profile shifts from around 530 nanometers into the infrared region, and infrared light will penetrate tissues more than light at around the 530 region. So what you can do with these materials is attach a targeting molecule, such as an antibody, to transport one of these shapes to a tumor. And if the tumor is not too deep in the tissue, you can use an infrared light source to heat up the particles. So the particles absorb the energy from the light, and it's essentially turned into heat. It can even cause rods to melt and turn into molten spheres. So obviously, if you've targeted your antibody conjugate to the tumor, you get a selective kill of the tumor when the infrared light is shone on the area. Okay, here's another therapeutic approach with particles. So this is usually spheres now. Again, same sort of idea. You target the spheres using an antibody or some other targeting mechanism to a tumor. The particles, once in the tumor, can be heated up. Now, at the moment, radiofrequency ablation, as it's called, involves inserting electrodes into the tumor, and then the radiofrequency therapy is used to heat up the probe inside the tumor. Obviously, that's not the particularly pleasant approach for the patient. So there's a lot of interest in non-invasive radiofrequency ablation, where nanoparticles, instead of electrodes, are positioned in the tumor by, as I said, some targeting mechanism. And radiofrequencies are very, very, very safe and easily penetrate tissues. It will go straight through the body and will only be halted inside the tumor by nanoparticles. So these particles will then heat up again by a process that's not completely understood, but has the same effect as the photothermal therapy with rods that I just showed you, except this is non-invasive, much better for the patient, and the tumor, whether it's on the surface or deep, the approach is just as useful for destroying the tumor. Okay, here's a summary of size and shape requirements. I'm sure you'll be able to find examples in the literature where sizes other than those that I've described here have been used, but this is just to give you a general idea. So techniques such as lateral flow, Western blots, dot blots, where it's a visual application, can essentially see what's happening by eye. Tend to use the larger sizes of gold particles, 20 to 80 range. Also, this range is used for dark field microscopy. I've not mentioned that at all today, but that's an interesting technique that uses gold nanoparticles. Electron microscopy, used for localizing antigens in tissues, in organelles. Generally, they're quite small nanoparticles, 5 to 20 range. Metal enhanced fluorescence that I showed you. Now, this depends on the electric field, and it's more intense the more rough or spiky the surface is. So this is perhaps one of the techniques where you don't want a perfect sphere. There's a lot of literature that says the enhancement is greater if you have a rough surface. So again, not really discussed this today, but there are methods that you can use where you can make gold particles with spikes. And there are other methods for roughening surfaces as well. The SIRS technique as well. Yeah, I think this one you definitely don't want a spherical particle. Very much greater enhancements if you have a rough surface. The fret quench approach, there's literature on particles of 5 nanometer up to 40 size with that. And again, I imagine you can find other examples with different sizes. The photothermal therapy and radiofrequency ablation, fairly new areas. Not entirely clear what the best size is yet, I think, for these techniques, or the best shape even. Drug delivery is obviously a massive field. Tends to be 40 nanometer or less because the larger sizes seem to be a bit more toxic. Can be rods or spheres in the case of drug delivery. Okay, well that brings us to the end of the presentation. I hope that's been useful, and I'll hand you back to Claudina.
Okay, thank you very much, Nick. Let's move on to our Q&A session. So there's still time to submit questions. Simply type in the questions box as shown in the picture on the slide. Thank you to everyone who submitted questions during the presentation, and Nick will answer as many as possible in the time allocated. If there are any outstanding questions, we will get back to those individually by email in the next few days. Okay, so our first question today is, can you coat any gold nanoparticle?
All the particles that we have tested so far, yes. That's 40 nanometer size from all of the well-known suppliers. We've also coated particles successfully from 10 nanometer to 80 nanometer in size.
Okay, we've got another question here. What is the best Raman dye?
We haven't done a huge amount of work on Raman, so I'm probably not the best person to answer this question. However, rhodamine 6G seems to be quite popular.
Okay, our next question is a disulfide as good as a dithiol for the self-assembly approach?
Generally speaking, thiols give tighter binding than disulfides, so if you want a stable attachment, probably the dithiol is better.
Okay, and here's another question. Can I attach oligos to innovaCoat Gold?
Yes, the easiest option is to use the maleimide gold derivative. For that you'll need a thiol on the oligo, but this can be added during synthesis.
Okay, and our next question is, can the one-step conjugation kit be used with proteins, not antibodies?
Yeah, I did say in the talk that the kit was for antibodies. It can be used with proteins. We've done the optimization for antibodies, but as long as your protein has lysines, then in principle, yes, it can be used. You may need to try different amounts of protein to optimize the system.
Okay, here's another question. I make my own gold. Can I buy your coating material?
We don't sell the coating material at the moment as it has to be freshly prepared. However, we can coat your particles in our custom lab if you're able to provide the particles.
Okay, our next question is, do you have to do covalent conjugation at different pH values?
No, you don't. It seems to be very different from the passive method. All reactions are done using exactly the same conditions. The only variable is how much antibody you add. But we provide advice with the kit. It's fairly predictable. Generally, you just try 3 amounts.
Okay, and I'm afraid we only have enough time for one more question. So our last question in the Q&A session is, can you provide bulk quantities of coated gold, or do you just have a kit?
Yes, we can provide bulk. We generally coat 50,000 OD units of gold at a time. And we can freeze dry vials at any scale. So the kit, if you like, gets bigger, but the process stays the same. You just add antibody to the freeze-dried mixture.
Okay, so thank you very much for sending in all those questions. Now please bear in mind that this webinar is recorded, so we will upload it to our YouTube channel in the next few days so that you can always come back to it. And you can find our YouTube channel at youtube.com/Innova Biosciences. Please keep an eye out on our social media channels where we will soon be advertising our upcoming webinars. And if you would like to receive a PDF copy of the presentation or a link to the video recording of the webinar, then please contact me. You can find my contact details on any of the webinar emails that you've received. So thank you very much for joining us today. We hope that you enjoyed the webinar, and we hope that you will be able to join us for our next webinar in October.